Little barley oats for the early afternoon. Hello, people. How is everyone? You should honestly start giving extra credit for every minute. Oh, snap. Who is this? Anthony Estevez. Oh, I don't know. I don't know. All right. So, with no more ado, why don't we just go ahead? All I'm going to do is go through the PowerPoints and kind of review what we went over in this fourth section. This morning, the reason I was 2 minutes late, no, not really. I just had to respond to this comment on Facebook. That's why I was late. I'm sorry. So what I did this morning was I put all the notes into one Word file and corrected the typos and things. So it should all make more sense. Can we go over chapter 8, slides 13 to 23? There's no recording of that. It would be great to revise it. Okay, so why don't we start there, chapter 8, and then we'll do the rest of it perhaps if we have time. Chapter 8. Okay. So this will be the second lecture in chapter 8. It begins with chromosomal packing. Okay. Let me pull my notes. Notificas. You can hear, but my dog is snoring behind me. I love him. He's got a hard life. Okay. Okay, chapter 8, lecture 2. So here in chapter 8, we're talking about how genes are controlled, how they're regulated, and how that relates to development. So we start out talking about packing of the chromosomes, because that's kind of the first level of regulation of genes, which is, you know, how compact or spread out the DNA is, because when it's compact, the cis regulatory regions are not accessible to transcription factors, so you can't get transcription from the genes. So we've got heterochromatin versus euchromatin. Heterochromatin is tightly packed and inaccessible, and euchromatin is opened up and accessible. And what determines whether that's open or closed is the histones and regulation of the histones. So some of the things that happen, probably the most important one, are methylation and acylation of the histones. They can also be phosphorylated, and that's important too. So methylation is kind of weird in that, like, acylation always opens up. Methylation can open up or close up depending on which amino acids in the little tail of the histone gets modified. So you can see there's a lot of different modification sites in the histones. And down here I talk about how, like, this methylation on histone 3, lysine 4 with one methyl group on it versus histine 3, lysine 27 with 3 methyl groups on it. So this shuts it off, and this turns it on. So it can be, methylation can go either way. So anyway, your genes in general are grouped into these topologically associated domains, which means that whole domain is going to be regulated in terms of opening and closing the DNA at the same time. So genes tend to be clustered in those things, in the sense that genes that need to be turned on all at once will be close together there. and you've got these little regions between where a protein called CTF will bind to help to open up that gene segment. Another form of control of the gene is methylation of cysteines. Cysteines in a CG grouping, and when you methylate that, it shuts that particular part of the DNA off. So basically, transcription factors won't bind to it, so now it's shut off. So this methylation happens because something changes in the environment that then changes the regulation of the gene, and then that can be inherited. That methylation pattern can be inherited, and this is why I always call it Lamarck's last laugh, because experience or life in general can change gene transcription and therefore change phenotypes. Okay, so now an example of how this DNA compression and stuff is controlled is in our HOX-A genes, the A cluster of HOX genes, which is kind of like right between two TADs. And what happens is when Wnt signaling occurs, when the cell experiences Wnt signaling, that activates genes at the 3' end of the cluster, and that causes, hang on, let me read it to make sure I don't get it wrong. Okay, HOXA1 enhancers are open because they're in between the two TADs. Then that begins the removal of the shutting off methylation and turns on methylation of the histone methylation that opens the gene. And then that kind of cascades down the cluster. And that's part of why we always see as you go from A to P, the HOXA 1, 2, 3, 4, 5, all the way down to 13 happens in that direction because the DNA is opening up in that direction. That's part of it. The other part of it, as you remember, was some transcriptional activators. I think it was CDX2 was turned on by the early HOX genes, and then that turned on the later HOX genes. So, but anyway, this is just an example of how regulation of the DNA condensation can control gene expression as well. And then the next thing we're talking about is so is this the chapter? Yes, this is the chapter when we're talking about differentiation. So going down a differentiation pathway. Why is it when a cell starts out as very general, it becomes a little more specific, and when that divides into two, the daughter cells are both that new cell type. And as they divide, if they're not interacting with any kind of other hormones, they stay in that cell type. Why is that? The inheritance of the pattern of gene expression. And one reason for that is because a lot of genes will cause their own transcription. So this is shown in this figure where this is MyoD, and when MyoD is expressed, it causes its own activation. It'll turn some genes off and some genes on, but it turns itself on. So so once the cell gets MyoD turned on, it will stay on in that cell forever, okay, until the cell terminally differentiates. And that's because, and part of that is because, you've got a cell with MyoD on, so you've got MyoD in the cytoplasm. When the cell divides into two, you still have MyoD in the cytoplasm of both cells, and that causes each cell to make more MyoD. So again, you've crossed that differentiation point. Now you've got MyoD on. Every cell after that is going to be influenced by that first induction of MyoD, because MyoD will always be there. Okay, so now with those general things in mind, we'll talk about how muscle tissue differentiates. So starting back here, which was chapter 5, you've got your somite, and the somite is separated into dermotome and dermomyotome. And then after, you get expressed to, or exposed to Wnt and Sonic Hedgehog and BMP4. This then differentiates into dermatome, this medial myotome and lateral myotome. So the myotomes here are then going to become myoblasts. So these get exposed to, I don't know 100%, but I think it's Wnt and BMP4, and that causes the upregulation of MyoD. So now you've got these cells, they're myoblasts, so they're still dividing and they're still mesenchymal and crawling. But when you remove the growth factors, they start to fuse to each other and then form a muscle fiber. So that, that really long multinucleated cell. And that's because of the interaction of the different transcription factors that happen at the different levels. So at the somite level, you get extracellular signals that cause the expression of PAX and PAX3 and PAX7, and that brings up MyoD, which brings itself up. So MyoD is now being expressed, and it also brings up MRF4 and MRF5, which also kind of express themselves. So this is what makes a myoblast, the fact that you've got these 4 different transcription factors here that are supporting themselves and eventually going to cause the cell to differentiate into a muscle. So when you remove your growth factors, then these cause the expression of myogenin as well as these myoFs, and that causes the differentiation to muscle cell. So there's an interaction here that we need to talk about between growth factors and these differentiation factors, because basically a cell will either proliferate or differentiate, one or the other, not both. And that's because of a direct interaction between the signals from the growth factor and the transcription factors that are involved over here. So growth factor pathway, we've talked about this before. If you're in my cell biology class, you're sick of looking at this, I hope anyway. I hope you've seen it enough. But anyway, when the growth factor is there, it activates this kinase cascade that then causes the induction of immediate early genes. Immediate early genes then make the late genes, the delayed genes, immediate early, delayed early genes. And delayed early genes include cyclin and CDK. So those are the ones that are going to cause the cell to go into cell division. But at the same time, and that's what I'm showing here, at the same time, okay, so here we are, cyclin and CDK causing the cell to go into cell division by phosphorylating RB. So E2F is the transcription factor that makes the cell divide. RB is a tumor suppressor that inhibits it. So when you've got cyclin and CDK around, it phosphorylates RB, and that causes the cell to divide. But now getting back to the differentiation idea, cyclin and CDK will phosphorylate MyoD, and so it gets degraded. So it doesn't totally disappear, it just keeps its level low. So it's constantly stimulating its own transcription and therefore translation, but as it gets made, it gets destroyed. So as long as you've got growth factors there, you're going to have cyclin CDK around, and that's going to cause destruction of MyoD, so MyoD stays at a low level. As soon as you remove those growth factors, though, you no longer are just degrading MyoD, so MyoD builds up, and then that causes the expression of MyoG, and then the cell goes into differentiation pathway. So that's kind of important. That's the interaction between differentiation versus cell division. And you can get one or the other, and they block each other that way. Okay, I believe that was Esha. My friend Esha asked me that. I went through it kind of quick. Is that, no, it's Ison. Is that good enough, Ison? I went through it kind of fast. Is there anything in that section you want me to talk about again? Ison? Ison? Thank you. Okay. All right. Well, in that case, let's go back to the beginning. Let's begin at the beginning, if you don't mind. And we'll start in that last bit of chapter 5, talking about neural crest cells, and we'll come forward from there and see how far we get in our hour and 15 minutes. I can keep going after that time, but you don't need to stay if you don't want to. Actually, you don't need to be here if you don't want to. I'm here for you, people. Okay. All right. Oop, somebody's chatting me. Chat. Come on, man. Chat. I can't see the chat. What is the, oh, there it is. Can you go through how regulation of the chromatin caused inheritance pattern? Okay, let me do that again because I just talked about that. And if it didn't make sense, we should do it again. Okay, so let me just do chapter 8 again, and then we'll get to that and I'll repeat it when we get there. So chapter 8 is all about differentiation and how cells become, go down a pathway to become differentiated. And that's this model here, Conrad Waddington. Is that an English name or what? Conrad Waddington. So each one of these kind of decision points involves hormone combinations hitting a cell. And when, depending on the hormone combination it sees, it's going to go right or left. And so if it goes left, it'll end up coming down this way. Then there'll be another decision point. It'll go down that way. So each of these decision points are all about hormones hitting it, causing different transcription factors to come up, and those activating different genes. So you've got this combinatorial gene transcription model. I'll get to that in a second. But let's see. Okay, so in terms of this gene control, you've got trans factors and you've got cis elements. So your trans factor is a protein, and it's called trans because the gene could be on any chromosome, but it's affecting this particular gene. And cis is because it's attached to the gene that it's controlling. That's kind of where the nomenclature came from. Before they knew anything about protein or DNA, they could see that they were attached or not attached to the same chromosome. But anyway, your cis elements, specific sequences of DNA, and they tend to be upstream of the core promoter. So the core promoter is the classic Tata box, and upstream of that are the control regions. And the trans regions then are, or the trans factors are proteins, and they typically have an alpha helix, and the amino acids, let me back up a tad, the amino acids sticking off that alpha helix then interact with the bases themselves of the DNA, and it gives you a very specific interaction between those two that means that the trans factor is only going to bind to specific sequences of bases. And you can see, you know, because each transcription factor has got like two different, what am I doing? Come on. Because each transcription factor has got, you know, two alpha helices, it's going, and each alpha helix is going to interact with about 3 bases. Then you've got about a 6 base pair sequence that each transcription factor will be specifically looking for. And so you've got several of these upstream of your core promoter. And what happens then is your trans factors will bind to these enhancers, and then you'll have co-activators sometimes also bound to these things by binding to that transcription factor. And then that brings this part, it helps to bring all of these other factors into that core promoter to start transcription. So these are what we call specific transcription factors, because they're only involved in specific, they're binding to specific regions in different genes. And then here are the general transcription factors that are involved in every gene because they bind to this core promoter. And then, you know, you've got activators and you've got repressors, or a repressor will maybe bind here and not let the co-activator bind so that it'll keep that gene off. So you can have different activators or different control regions on different genes that are put together in different combinations, which will give you different phenotypic phenotypic responses because you get different genes being turned on. That's the combinatorial control. So transcription factor can turn a whole bunch of genes on and at the same time turn other genes off. And so you get a specific combination of proteins being made that's going to then differentiate that down a specific pathway. So that's the, why am I doing this? I don't know. That's the gene gene control we usually talk about. Now to step back, like if this whole region of DNA is wound up with histones, then transcription factors can't bind to it, so it's going to be off. That whole section of the gene will be turned off, and that's what we're talking about here, where control of the condensation of the chromosomes can control gene transcription. So again, if you've got a gene in here somewhere, there's no way a transcription factor can bind to it. But if it's over here in this euchromatin section, then your transcription factors can bind to it. And that can be regulated by methylating, acylating, phosphorylating histones. You can see how many different potential spots there are on this arm sticking out of the histone to regulate it. Methylation sites, phosphorylation sites, acylation sites. It's like lysines and arginines are popular for methylation and acylation. Phosphorylation on tyrosines and serine, or no, threonines and serines. Okay, and then this is the example. So basically, genes that are regulated together tend to be, look at that, tend to be in a into all stuck into one region that is regulated together in terms of collapsing and condensing. And the example of that we talked about is the HOXA cluster, and the HOXA1 control region is somewhere between two different GADs. So that can get turned on by Wnt because it will be responsive to Wnt. But then once that gets turned on, it activates, changes the methylation of the chromosome, the rest of the chromosome in this 5' direction. So then it starts to open up other genes by activating that methylation change in the histones. Okay, what's that? Dared to ask that question. Can you go through how regulation of the chromatin caused inheritance of pattern? Okay, so that kind of had I think I just said that, and I know you asked it earlier, so, but let me just say it again to make it more obvious. So like with that one example of the HOXA genes, you know how HOX genes are expressed from the number 1 all the way through to number 13. So when one gets turned on, that causes a change in the condensation of the chromosomes to the 5' end. So now the 5' end genes get opened up. Now they can be transcribed, and then the further 5' end get opened up, they can be transcribed. So it kind of hops down the chromosome to allow them all to be transcribed, but only in that direction from the 1 to the 13 gene. Yes, I will post the recording. As long as it records, it is recording now. Okay, let's go, if we may. What am I doing? This one, escape. Discard that. Okay. 10, 7, 5. Where's 5? 5, 7. Okay. So All right, neural crest. Where do our neural crest cells come from? So, as you know, the neural crests come from cells at the dorsal side of the neural tube. They undergo that MET conversion. No, EMT conversion. They get converted to mesenchymal cells and they crawl off. Now, how the hell does that happen? Glad you asked, because I know the answer to that. So early on before early neurulation, your notochord changes some of this ectoderm into neural ectoderm, and that's under the influence of Wnt and FGF. And then a little bit later, we see that you've got neuroectoderm, but then these are the cells that are going to become neural crests. So why is that? Why is the lateral sides of this neural plate become neural crest? We'll talk about the signaling in a second. But anyway, that neural tube then closes up. Those green cells, the neural crest cells, end up at the top. And then this closes, and we'll talk about that whole process later in chapter 7. Once it does, these cells then undergo that epithelial to mesenchymal transition, and then they start to crawl, and they go off laterally, never anterior or posterior. They go lateral, they go ventral, they never go AP. And they make basically neuronal type cells for the most part. Your glia and your Schwann cells are associated with neurons, and they're support cells, sensory neurons, sympathetic, parasympathetic. These are parasympathetic also, or no, it's not, but it's neurons controlling the GI tract. And then the cartilage and melanocytes. So cartilage and melanocytes are not neural, but these are all the cells that these things become. Okay, so why is it we get neural crest cells? And that's because Wnt and FGF is making this ectodermal, but then you've got BMP4 coming from the lateral mesoderm, and we've got a gradient of BMP4. And so where you have no BMP4, you've got SOX2 being expressed. Where you've got high BMP4, you don't have any of the SOX genes expressed. But there where you've got weak BMP signaling, but you still got FGF, Wnt, and BMP, you're getting SOX9 and SOX10. And that brings up snail. And snail is going to cause that epithelial to mesenchymal transition. And by the way, I changed this today because I looked it up and it was wrong the way it was in the notes before, so I fixed that. Okay, and then as the neural tube closes, so as you get to like this stage, then you get slug and twist coming up. Because here, when this thing is closed, remember you've got BMP coming from the side, right? And you've got Wnt coming from underneath, like all around here. So you get a really high concentration of Wnt and BMP4, and that brings up slug and twist. And slug and twist then also help with the crawling and the neural cresting of the cells. Okay, so this is mainly just, now we're mainly talking about the fact that these cells are only going to be whatever they were supposed to be in their position on that AP axis. And that's again Hox genes. You know, here your first few Hox genes come out even before the somites are being formed. And then all the rest of the Hox genes come out later. But you can take, if you take cells from a different area on the AP axis and move them, you'll find out that they're still pretty multipotent, but they're still designated to be in that position of the AP axis. So here we are, looking up in the brain. So these are rhomboids. So somites are mesendermal segments going through the body. Rhomboids are segments of the neural tissue going through the body. So these are brain rhomboids, and then these are spinal cord. So this is basically neural tube segments. And what they're showing is that, you know, rhomboid 2 will always become what it is, jaw, and rhomboid 4 will become throat, okay? And again, it's neural crest, so it starts out as neural tissue and then crawls ventrally and then becomes throat. But you're still, because of that Hox gene expression, these cells start out as just general neural crest cells and eventually differentiate into, the mesenchyme of the throat, or the mesoderm of the throat. But, because there are 4, they will always be throat. So if we took this and put it down there, you'd end up with the throat further back. If you take number 2, which is supposed to be jaw, and put it in place of 4, you get 2 jaws instead of having the throat. So you do have this regional specification based on the Hox gene expression. That's all that's trying to say. Left, right. So mostly your left side and right side, at least externally, is symmetrical. In fact, that's one of the things they say is important in physical attractiveness. Like if your face is not symmetrical from one side to the other, you're considered unattractive. Although we all have slightly different shapes from one side to the other. If you like cut a picture of yourself in half and flip it and put it together, it doesn't look like you anymore. It looks pretty different. So there are some slight subtle differences externally, but internally there are big differences, right? Your liver's on the right, your heart's on the left, your spleen's somewhere there, wherever the spleen is. Why does that happen? Has a lot to do with nodal. So here in mouse, this is like the bottom of the node during neurulation. That node is moving during neurulation. Right on the bottom of it, you've got these modal cilia. And then you've got other cilia out here that are not modal, they're more sensory. And so these cilia at the bottom of the node are constantly swishing to the left, swishing to the left, swishing to the left. And what that does, a couple of things, possible things anyway, that does, is it's activating these sensory cilia here that are causing these cells to release more nodal. Okay, so this is giving off a signal transduction. It's causing more, that's causing calcium release and more nodal release over here. And also it's kind of grabbing some of this nodal and swishing it over to there. So higher nodal on the left side makes that into left side. Lower nodal on the right makes that on the right. And then in chick, it's different in chick. That was mouse and probably human. In chick, it has to do with a gradient of hydrogen potassium ATPase activity. So you've got more HK ATPase activity on this side. So as a result, you get calcium release over here, and that causes nodal release over here. So you've got nodal being, before nodal, notch. Serrated and jagged are I think it's jagged, are ligands for the notch receptor. And they get expressed because of that calcium, so you get notch signaling. Notch signaling causes nodal signaling. Sonic hedgehog signaling also helps to increase nodal signaling. And this high nodal signaling causes an upregulation of the transcription factor PITX2, and that's going to make this left. And this is going to be exaggerated, I guess you would say, by activin, which is blocking sonic hedgehog activity over here. Okay, so we're getting a lefty expression here, and that's making this lefty. Lefty, okay, so that's the end of chapter 5. Chapter 5. That's a very Vermont way of saying it, 5. Okay, questions about chapter 5? Shout them out. I can't see my chat box. Nobody knows. Okay, good. So, chapter 7. Pull up my phone so I know what time it is. Okay, so chapter 7 is all about, it's not about at all about signal transduction or transcription or anything like that. It's all about cell movements and cell changes, which is based on the signal transduction, but it's downstream from that. Oh, we have a chat. Can you just shout out your questions instead of chatting them? Because when I'm like this, I can't see my chat. Whoever chatted me, yell it out. Por favor.
I was like, what did you correct on the slide again?
What did I correct on this slide?
No, on chapter 5.
On chapter 5? The one thing I changed was this. This is the slide I changed because previously I said that this caused the upregulation of slug and twist, and it doesn't. It causes 9 and 10 to come up, which causes snail to come up, and snail causes EMT, and then later you get slug and twist being expressed. Okay, and those are all transcription factors.
Okay, thanks.
Okay, so this whole chapter, a lot of this chapter is all about how cells grab each other, how they hold on to each other, and how they hold on to extracellular matrix. So we've got two different types of connections that are cell to cell, and cell to cell connections are all about cadherins. Your cadherin cell to cell connections are usually same cadherin bind to the same cadherin, which is homophilic, right? So these homophilic junctions then are called adherens junctions. I guess I should talk about that here. So here's your adherens junctions here, and they connect to the cell, to cytoskeleton, and they activate signal transduction. So this is doing it, these are holding same cells together like in a tissue, and it's also signaling to the cell that it is attached to the same cell already. So, you know, cells don't have eyes. This is what allows them to see each other. So there's signal transduction going on in these adherens junctions. Desmosomes are then when you've got different cadherins attaching to each other. That's heterophilic. And this is doing the same thing. It's attaching to the cytoskeleton and keeping things together, and also giving signal transduction. Here's just another figure of that. Okay. Oh, another difference here. I think these desmosomes are attached to intermediary filaments instead of the cadherins are attached to microfilaments. I guess that's not that important, but it's a difference. Integrins then are attaching cells to the extracellular matrix, and this is really important for cell crawling. Right, we'll use that during cell crawling, but also even when cells are in tissues, they're attached to the extracellular matrix, fibronectin especially. Okay. And that again attaches to the cytoskeleton through microfilaments, and you've got signal transduction going on in both directions. This tells the cell that it's attached to extracellular matrix, and this and the cell can then tell it to grab it or let go of it. So, you know, you can talk in either direction here. And so we talked a little bit about cytoskeleton, microtubules, microfilaments, and intermediary filaments. Microtubules are important during interphase and mitosis, and you've got motor maps that walk along the microtubules to give you movements during movements based on microtubules. So way back we talked about xenopus fertilization and then cortical rotation. That cortical rotation was caused by microtubules and motor maps somehow. Not explained specifically. And then your cilia are also moving because of motor maps. They're microtubule based and moving based on motor maps. Microfilaments then are made of actin. They move based on interactions with myosin, just like in your skeletal muscle. And you've got three different arrangements of these microfilaments. You've got anti-parallel stress fibers, which will pull the cell forward. You've got gel mesh, which is part of your lamellipodium. And then you've got parallel things up here in the filopodia. And I've never really figured out what filopodia do. They're always important, but I don't know what they do. Anyway, here's your gel mesh for this red on the outside is the cell cortex supporting the plasma membrane. And here's our cell crawling, which our mesenchymal cells do. So they reach a lamellipodium out, make a new focal adhesion, and then the anti-parallel stress fibers pull the cell forward towards that new lamellipodium, and then the lamellipodium just keeps extending out. Okay, EMT and MET. So this is transition back and forth between connected cells in the tissue and mesenchymal cells that are loosely connected to each other, if at all. And we see this happening a lot in embryo development. When during neurulation, when cells come to the node, they undergo EMT and they become mesenchymal, they move, and then they become part of the somites and they undergo MET. They go from mesenchymal back to epithelial. So this happens a lot. Adherens junction. So this is kind of an interesting experiment, and it helps us to understand the compaction of the morula in the next slide. So basically, if you take cells that are ectodermal and cells that are mesodermal, divide them both or dissociate them all up into cells, mix them all up into one big conglomeration, they will separate themselves. And why is that? Because so you've got a ectodermal cell that's surrounded by mesodermal cells, and then suddenly hits another ectodermal cell, boom. It makes cadherens junctions and attaches to itself. And then another one accidentally falls into it, boom, now you've got three, and you're nucleating and you're getting bigger and bigger. So these ectodermal cells are not going to attach to the mesodermal cells because they've got different cadherens. And if they're allowed to freely move around like this, they will eventually sort themselves so that it's only ectoderm with ectoderm and mesoderm with mesoderm, because that's the only thing that can happen because of cadherens junction or adherens junctions. Okay, how does that help us? So here in compaction of the morula, which is really important, early in 8-cell stage, they're kind of all separated from each other. It's eight individual cells, and then they start making these adherens junctions, which makes them all grab each other and pull in closer. So you've got cell-cell connections that are pulling these cells together more. And that, as you know, affects the expression of CDX2 and OCT4, because when you've got a free apical surface, the hippo pathway is off, and so you get CDX2. And when you're on the inside and all your surfaces are connected, hippo pathway is on, and you get OCT4. So these adherent junctions are really important for that compaction of the morula. Also helps to keep cells into a coherent sheet. So in this Xenopus gastrulation, you've got a sheet of cells here that are a combination of mesodermal cells that are attached to each other with adherens junctions, endodermal cells attached with adherens junctions, and then desmosomes between the endoderm and mesoderm, okay? And that's keeping that as one coherent sheet. And there are no connections, though, between the mesoderm or endoderm and this ectoderm. So this sheet can move along that sheet because they're not grabbing each other. So those cell-cell contacts are really important in this gastrulation. And the gastrulation itself is all about convergence and extension. So these cells are not dividing at all, okay? But what they're doing is intercalating with each other. So as they go, this is lateral intercalation. It's kind of like Play-Doh, squishing the Play-Doh together, and it's getting longer. So when the cells, you've got cells like this, and they do this, and now they're longer. The tissue is longer. So as the cells interdigate with each other, the tissue gets longer. Convergence and extension. And this is a really freaky part, the way you've got the blastopore over here, and you've got mesoderm all the way around and endoderm under it, and it all gets pulled, cell to cell pulling up towards that blastopore, and then gets shoved through the blastopore and elongates as it goes through that blastopore. And and that's what your gastrulation is. And cells that are near each other in the blastula can be pretty far away from each other in the neurula, okay, after gastrulation has happened, because these cells are crawling faster than these cells, so they get pulled through that blastula sooner. And then these things are coming in behind. And because you've got the two sides coming together into that blastula, there are some cells that are far apart in the blastula, and when they come to the blastopore, they end up close together. So where you are when you're in college is not where you're going to end up. I don't know what the hell that means. That was kind of weird. I don't know where that came from. But anyway, this is showing that again. Here's A and B are close to each other. I guess A goes through first because it ends up way anterior, and B ends up way posterior. So they get separated, and they stretch that mesoderm out again by interdigitating and convergence and extension getting stretched out. And the mesoderm from the sides gets pulled together into somites that are opposite each other. Convergence and extension. Epibole is also interdigitating, but it's on the, or intercalating, but it's on the radial axis. So your blastula's got a thick layer of ectoderm. Okay, these are inner cells and these are outer cells. These outer cells go up into those inner cells, and it thins out. So it goes from thick to thin, and as it thins, it spreads around the embryo. Because where else is it going to go, right? It converges down around, and it comes to the blastopore. Radial intercalation and circumferential expansion. Who wrote that? Oh, I wrote that. That's pretty smart. Chick gastrulation also involves convergence and extension. So these ectodermal cells from the outside are converging on that primitive streak. You know, they're coming together, and then they're going in, and then lateral and anterior to that. So you've got the convergence of the cells at the primitive streak. And once they get there, you get shape changes. So cells become, you know, the apical side squishes in, and the other side squishes out. So now you've got a differently shaped cell, and that causes it to form the groove of the primitive streak. And then once they get here, they get pushed inside, and then they undergo EMT, and they become mesenchymal, and then they move laterally and anteriorly from there. Okay, so again, what this is trying to show, what's important about this in terms of chapter 7 is EMT, okay, letting go of those cadherens junctions, convergence and extension, because these epithelial cells are being pulled in this way. And what else did I want to say? Oh, shape changes, because the cells are changing shape, which is causing them to push inside. You can kind of see the progression here from a cell with a wide apical section to this one's got a really narrow apical section to this one is like inside the primitive streak now, because that apical section totally went away. And then it becomes mesenchymal cell. Okay, so this is talking about notochord in the Xenopus. Go back to Mr. Xenopus. And you can see how, like, this is the notochord now, okay? Hasn't gone anywhere. It's not notochord yet because it's pre-gastrula, but it's like this long. Over here, it's this long. So you get that extension because of changes, cell shape changes as it converges and goes in. So that's what this is talking about. This is kind of that first picture. And then the cells intercalate, and it gets longer, and they change shape. And then the cells change shape again, and it becomes kind of this cuboid, or not cuboid, kind of this shape thing. And then it continues to change shape. And as they're changing shape, the cells are intercalating and getting longer and extending that from that little squishy little area there by the blastopore out as the gastrula extends to become the notochord. It's beautiful. It's beautiful. Shell shape changes again here, causing the neurulation, the rolling up of the neural tube. So you've got underneath this ectoderm, you've got that notochord releasing FGF and Wnt, turning this into neuroectoderm. And then these cells at the bottom, you get apical constriction, so that the apical side of these cells constricts and the other side is going out. So that forms a wedge-shaped cell. And that wedge-shaped cell then brings the sides up. And then as the sides come up, then you get a second shape change here, dorsolateral hinge, and that brings the sides together. So you've got the first hinge bringing them up, the second hinge bringing them together. And then you've got cells up here that have got lamellipodia sticking out. Those are the ectodermal, or the epidermal ectoderm up here. Lamellipodia stick out, and you can imagine lamellipodia kind of going past each other, interdigitating and closing up that ectoderm on the top to seal the neural tube and seal the ectoderm on top of it. And this is what happens if it doesn't seal. Yuck. Okay, different cell type changes, or different things going on in this chapter. Your cell crawling, lateral intercalation, giving you extension, radial intercalation, causing it to flatten out and spread, and ingression, when a cell undergoes EMT, it lets go of its external cells and then it goes in. And then shape changes, which are going to cause extension as well, or change in the shape of structures anyway. That's morphogenesis. And last but not least, let's make a limb. Let's make some chicken wings. Let's make some chicken wings. Chapter 10. I don't remember this. I don't remember this. Okay, so all I'm trying to say here with these two are, this is where the muscle cells are going to come from that will fill that wing. And that's this lateral myotome. And this is again talking about regional specification, because that becomes an issue with whether you're going to be a wing or just a thorax or a leg or a tail, right? Because your Hox genes are determining that. And that comes into play in this chapter as well. There's what arms look like if you've never seen one before. Okay, so this process all starts here with this apical ectodermal ridge. So the apical ridge, thickened ectoderm, and we're going to get expression of FGF8 from that, and that's going to help with this extension of these limbs. Here's what it looks like in the end, blah, blah, blah. Oh, this is just about, I think the reason I put these in there is just so you can see what I'm talking about when I say anterior, posterior, dorsal, ventral, proximal, distal. Proximal, distal, the front part of the wing is the anterior, the back is the posterior. Then you've got the top, which is dorsal, and the bottom, which is ventral. We didn't get into dorsal ventral because I ran out of gas on Monday. Didn't get to that. I'll do it next year. You can take the class again next year. I could fail you all. You can take it all again next year. Wouldn't that be fun? No, it wouldn't be fun. Okay, so apical ridge is out at the end, and that's ectoderm. And then inside of it, you've got the zone of proliferating undifferentiated cells. So we're making cells here, and that's causing the limb to extend. Behind that zone, that ZPA, zone of proliferating activity, you've got cells that are differentiating into cartilage and then eventually bone. So these cells are going to be cartilage and bone. The muscle for this is coming from those that lateral myotome, and then the nerves are going to come from neural crest cells that will migrate out here. And here's the regional specification. So if you're up in here, let's see, do we have another one of this? There. So if you're up in the upper thorax here, you've got Hox gene 4 and 5 being expressed, and that brings up TBX5, which tells you this is going to be a wing. So TBX5 is telling you it's going to be a wing. TBX5 brings up FGF10 in the zone of proliferating activity, ZPA, and FGF10 then goes to the ectodermal ridge and causes the expression of FGF8. So you get this mutual admiration society here between FGF8 and FGF10, and all that FGF causes proliferation and cell movement. So that causes the limb to go out. And then if you're down here with, you're down here further, you've got Hox genes 8, 9, and 10 being expressed, and that causes TBX4, and TBX4 then is going to cause the same thing to happen over here, but this is definitely going to be leg and not wing. And you can see that from this experiment where they take, and this is interesting. This tells us two different things, this experiment. So they take a bit of this mesoderm from what would be like the thigh of a leg and put it up where it would be the hand of a wing, and you end up with still, leg specification because you're getting toes and not arms, but it's not a humerus, it's the hand. So this is telling you that you've still got when you take things from down here, you've still got your AP designation done, but you still have some, it's still multipotent in that if it stayed here, it would have been thigh, but because it's on the outer edge here, it becomes the distal part of the wing. Okay, so we're learning two different things through this experiment. That might be a good question on the test. Two things do you learn from this experiment? Okay, and then we also have our ZPA, zone of polarizing act. I'm sorry, I've messed up. I'm sorry. I've been calling this the ZPA. It's not. This is zone of proliferating cells. So the P in ZPA is zone of polarizing activity. So this little spot at the posterior end of the limb bud is the ZPA, and it's releasing sonic hedgehog, which is going to give us this AP patterning of the limb. This is an interesting experiment too. I'll come back to that. Let me talk about the AP patterning of the limb. No, let's not. Okay, let me back up. I'm sorry, I know I'm jumping around a lot because I'm so excited about this. Okay, we talked about that. Okay, so this, backing up a bit, this is what causes the extension of the limb bud, this expression of FGF10 and FGF8 that are reinforcing each other. And here's the experiment that shows that. If you take the apical ridge off an early limb bud, you only get the more proximal structures. If you wait a little longer and you take the apical ridge off, you get a little more leg. And if you take it off late, you get a little more of this wing. So that's what's helping the thing to extend, is that FGF coming from the apical ridge, which is being caused by the FGF coming from the proliferating cells. And here it's shown again by putting a bead, taking off the apical ridge, but putting an FGF8 soaked bead on that end, and that causes it to extend almost normally. So FGF is really important here in that extension. So here's kind of chapter 7-ish things that are involved in limb bud initiation. So normally your cells are aligned in this anterior to posterior direction next to the somites. But under the influence of Wnt, some cells will turn sideways and start going perpendicular to the way they were going. And then they all kind of turn in the outward direction. And that helps the limb to extend because they're crawling as they and proliferating in that direction. And that gives you the extension. Okay, so now, so that was just talking about how the limb extended out. How do we get from here out to here? Now, why is this different from this, different from this? How do you get patterning from the proximal to the distal end? And there are two different models for that, and they're both right, basically. The first model is that in this progress zone, where you've got the proliferating cells, there's like a cyclical clock going on, which the transcription factor HARI2 is involved in. So you've got a cyclical coming up and going down of HARI2, and so cells will experience that. That's the timing model. So cells that only saw a few cycles of that clock will end up being proximal. And the cells that have seen the most cycles of that clock end up being distal. And we know this clock model works in other places, like in the node during neurulation. So we know these clocks can work. So this is one model. The other model is the two signal model, where you've got that FGF coming from the apical ridge, which is going to oppose retinoic acid coming from the trunk, okay, the main part of the body. And we know again from the pre-Semitic mesoderm that these two hormones inhibit each other. So you get a strong gradient from RA to FGF when you've got them both expressed there. And so these gradients then determine the proximal to distal patterning. Okay, so because things that are very proximal are seeing mostly retinoic acid, things that are distal are seeing mostly FGF, and things in the middle are seeing a certain concentration of both. And what they come up with in the end is kind of a fusion of these two models, where early on during limb bud initiation, you've got the two-factor model where you've got this gradient of retinoic acid versus FGF, and that's starting to pattern the proximal parts. But as this progress zone extends out, the cells stop seeing retinoic acid. And so once you get past that retinoic acid period, now you're on the clock model, okay? And so your AP patterning is based on that clock. Okay, that's kind of complicated, but I think it's neat. And this is pre-Semitic mesoderm, but it's just showing you how the clock works with FGF and Wnt and Notch. And so this is HARI1 in the limb, but it's HARI2, but it's a similar kind of a thing with the thing. And now here's your AP patterning, which is coming from that ZPA, zone of polarizing activity, and it's releasing Wnt. So you've got a hedgehog. So you've got a gradient of hedgehog, and that's what's giving you the difference between the posterior side and the anterior side of the wing. And here's some experiments that show that. Take some of this area from a different embryo and put it out here, and now you've got two gradients of hedgehog, and so you end up with a twin wing. Okay, so you've got your posterior side over here, which is being induced by this, and a posterior side over here, which is being induced by the endogenous ZPA. And it's definitely a gradient because there's a dose-dependent effect. If you put fewer of these cells over here, then you get less hedgehog being released. So your gradient is less over here. So you get less of a posterior side over here. Oof, and that's it. What time is it? 2:07. Oh, I guess this class ended 15 minutes ago. Oh, well. I gave you a lot extra there. Any questions? Not too many people left. Stefan's here, Rocky, Elijah, Lauren Bintz, Alex McCurran, and Rhonda. Okay, well, I'm going to let you go, and I will post this, and you can watch that again. Thursday will be our final then, and I'll probably get it back to you early next week. Well, I hope anyway, by Tuesday. All right, guys. Talk to you later.